It is a great pleasure today to present the webinar. We are going to hear something a bit different, but I think that this is in our future as well. Mario Rossi, he always says that he is really called Mario Rossi. It's not let's say the classical Italian man. But anyway, Mario Rossi graduated in the University of Buenos Aires and did the PhD in the University of Rome with Gianni Cesareni. Then has done postdoctoral studies all around the world, in Leicester, UK, in the MRC, in Haifa, in Israel, in the Technology Institute, and in the New York University Lange Medical Center. And then he went back to Argentina in 2012, so 10 years ago. There was a new Max Planck Institute for biomedicine in Buenos Aires. He joined that and developed a program on autophagy and ubiquitin modifications. In 2018, he moved to the Austral University, and he is now there, professor of biomedical engineering, and he also is professor in the Hospital Italiano di Buenos Aires. He is now involved in artificial intelligence teaching, and he is going to tell us now a little bit of his new orientation, because he did in 2020, I think, a master on informatics, and he has dedicated to this subject since then. Mario?
Okay.
The floor is yours.
Everything is looking fine. Okay. So first of all, thank you all for and first of all, thank for the invitation. Oh gosh. Thank for the invitation. It is a pleasure and an honor to be here today. And I will try to in the next couple of minutes, I will summarize some of the work I did in the past, and at the end we switch gear, and Tito told you I will tell you what we are doing right now. So for many years, I've been working on a concept similar to homeostasis, but on proteins. Therefore, checking the proteostasis and checking different pathways that are related to that. Basically, there are two big pathways relating to that, which is ubiquitination and autophagy. Both of them are very complex network of enzymes that need to act in a concerted action, and actually those are only maybe only the most complex post-translational modification is glycosylation of protein. And it's not only one thinking in ubiquitin will always associated with the protein degradation, but that is only half or just a part of it. Autophagy is also a very complex system, which basically is forming membranes that are eating discrete part of the cytoplasm and taking it to degradation. So it's fair to say that both systems regulate almost every cellular process in a way or another. So and that is not surprising that many genes associated both to ubiquitin or to autophagy the regulation mutation or different expression is associated with different pathologies, including cancer. And to make things even more complex, there is a crosstalk between the two systems where ubiquitination is somehow talking and resembling with the crosstalk when the different signal transaction cascade were discovered, and then they were talking about crosstalk. This is something similar, but being a little bit more complex, given the fact that these are very complex post-translational modifications. And so for many years, what I did is based on the fact that in a normal cells, ubiquitination and autophagy are important for, as many others, pathways are important for the life of the cells, and they are always found disrupted or modified in cancerous cells. The idea was to study this pathway to see, first to understand what the role they were playing in this, in the tumor homeostasis, and how you can modify it in order to gain a better understanding of the tumor biology. So long time ago, I started with a project that was basically dealing with P53, this tumor suppressor that is very well known, and that is most of the time is mutated in cancer cells. And the idea was one way to reactivate P53 when P53 was not present. There is P63 and P73, and one way of activating is instead of checking on phosphorylation or other modification, is increasing the amount of the protein that is present. So with that in mind, what we did is we found a E3 ligase that was degrading P63 and P73, and we saw that when the E3 ligase was inhibit, the levels of P63 and P73 were increasing and leading the cells to go to apoptosis. So what we try to do is looking for any pharmacological inhibitor, and we performed a screen, a high-throughput screen a long time ago, where we, among many other things that were in the library, we identified a whole family of tricyclic antidepressants that were working on modifying and acting on each. The surprise was that we demonstrated that these compounds are acting on the are binding and are modifying the function of this E3 ligase, and actually in collaboration with Simona Polo, then she did further works to understand exactly at the molecular level how these molecules are working and how they are inhibiting the molecular action of this type of enzymes. But what was interesting is that when we were looking at this and we're treating the cells, what we figured it out is that by looking more carefully, when we were treating the cells, although the cells were completely healthy and they could live without any problem and divide without any problem, they were developing these tiny vacuoles inside the cells that were kind of somehow representing some kind of stress, although the cells could endure that stress without any problem. And that led us to speculate whether this type of tricyclic antidepressant were involved with the regulation of autophagy. So basically, we did a couple of papers and experiments showing that indeed tricyclic antidepressant modulate the autophagic pathway, and that would be important for the repurposing of tricyclic antidepressant. First of all, explaining some of the effect that these old drugs have, and more importantly, maybe reposition these drugs for other purposes, like in cancer or other type of diseases. So, but more recently, and since a couple of years, we tried to move and did another type of screening. And in this case, we figured we wanted to look at features that were related to aggressive cancers. And therefore, we focus on metastasis. And to have a proxy of metastasis in vitro, we focus on motility of tumor cells. And we knew that ubiquitin had a role in controlling migration and invasion of tumor cells, and therefore ubiquitin pathway was an attractive target to work with, and we decided to look for new targets in that pathway. To do that, what Fabiana did is actually she developed a screening where she infected the cells with shRNA against genes related to the ubiquitin protein system, and using a sequential and enrichment cycles, where what we did is infecting the cells, then allowing them to migrate through the trans wells, and removing the cells that had migrated through the trans wells, and amplifying the cells that didn't migrate. And by doing that, enriching the cells in those shRNAs that were inhibiting the motility of the cells, and therefore, if enriching in shRNA that were targeting possible positive regulators of migration and invasion. So after the several cycles of selection, what we got is that the control cell line that was infected with the empty library did lose a little bit of migrating potential, but not as much as the one where we infected with the library containing the shRNA against the ubiquitin path. Okay. And among the targets we found, what we did several type of recursive analysis, and we tried to figure it out, or to analyze genes where nothing was known before, or there was nothing connecting it to either metastasis, cancer, or migration. And having after the selection cycles, we had at least 60 target genes. 50% of them were known targets, given, so they were a proof of principle indicating that our screening had worked, and therefore we focus on the other one that didn't show or were not known to be connected to metastasis, and we chose this gene, which is called USP19, which instead of being of adding ubiquitin to the protein, is removing. So if you think in terms of kinase and phosphatase, this family, or USP19, belongs to a family that would be more close to kind of phosphatase in terms of removing the mark that the other system is adding. So no many things were known at the time. Just a couple of days ago, it came out a paper showing that USP19 is playing a role in hepatocellular carcinoma. But at the time, nothing really was known. There were scattered papers all around about the function of USP19. So what we did is, when we inhibit the USP19 using 2 shRNA, what we saw is that the cells were changing the shape from a mesenchymal type of cells to more epithelial cells, and that doing wound healing assays, when we are inhibiting USP19, the cells were taking much longer to close the wound, indicating that they had a problem in the motility of the cells. Similarly, what we did is, instead of looking at the just migration, we look at invasion, and we used a system where we had an agarose bead where we see the cells around it, and we could track live the cells, how they were entering the agarose bit. And what we saw is that the cells couldn't enter the bit as compared to the control cell line. So basically what we found there is that the USP19 was involved, and the genetic ablation of USP19 was causing impairment in cell motility and in cell invasion, while it was not affecting cell survival, at least in 2D conditions. So then we moved to a more in vivo system. Well, the idea was to inject the cells where USP19, the control cells, and the cells where USP19 was knocked down, and see whether the mice could develop or xenograft transplant tumors. Or the other system that we used was injecting the same cells through the tail vein and assessing the metastasis after a couple of months of injection. What we observed is that basically when we knock down USP19 using any of the two different shRNA, basically the tumors didn't grow, or almost didn't grow compared to the control one, and the incidence, meaning the amount of mice that developed the tumor, were almost reduced at half, and the ones where the tumors grow very slow. So not surprisingly, also what we observe is that when we inject the cells through the tail and waited at least a couple of months or so, and we dissected the mice, what we observed is that the amount of cells that were colonizing the lungs, this cell line is known to go and colonize the lungs. And actually what you see here in this, in the lower panel, is that you basically dye the lungs with Indian ink, and the white spots are the micrometastases and the metastases in the lungs. What we observe is that knockdown of USP19 reduced significantly the amount of metastases that appear in the lungs. And to have a more accurate way of measuring, given the fact that we injected human cells, we measured the ratio between human DNA compared to mouse DNA in the lungs, and we have the confirmation that actually not only the micrometastases, the one that we could see, but also the micrometastases were highly reduced. So in order to make the story and prove it a little bit further, what we decided is, instead of taking cells that were migrating very fast and knocking down USP19, we took cells that were not migrating. These cells are MCF7, are also a breast cancer-derived cell line, and we overexpress USP19. The idea was that also USP19 has a catalytic activity, and we had the possibility of overexpressing the wild-type version or the catalytic dead mutant, because most of the time in this, when you're dealing with genes and you're deleting the gene, among other possible problem that you may face is the fact that sometimes it's not related to the catalytic activity per se. So we wanted to test this, and what we observed, the first thing that we observed is that by overexpression, maybe once you look at the cells for long enough, you can see a difference. Maybe here it's not that obvious, but the wild type cells look much more elongated and spread around compared to the parental one, which is the one we expected, and the one that were expressing the catalytic dead mutant resemble the parental one and not the wild type, meaning that if anything, it looked like the catalytic activity USP19 was required for whatever function it was doing inside the cell. So we had on one side, when we were taking out USP19 in cells that were able to migrate, they stopped migrating, and we overexpressed USP19 in cells that were not migrating, the cells start migrating and moving or looking at more mesenchymal. Actually, we did the same experiment on and I will not show because it was published. We did all the experiments on migration, and and then we repeated the experiment on xenograft transplant. It's important to say that these MCF7 cells cannot, normally they don't grow, they don't produce tumors if they are not supplemented with estrogen pellets. You have to put estrogen pellets subcutaneously in the mice to provide enough signal for the cells to start invading and producing the tumor. So if you don't put the pellet, normally these cells don't grow. So what we did is we injected the 3 cell lines, the parental one, the one overexpressing the wild type version of USP19, and the one expressing the mutant version of USP19. And what we observed is that, as expected, the parental one didn't grow because we didn't put the estrogen pellet, but only the wild type grew. So this is relating to the fact that somehow is overcoming the and producing a more aggressive phenotype that can somehow be independent on the presence of estradiol in the bloodstream. Then doing some bioinformatics, what we found is that the genes that were mainly associated with overexpression and that were differentially expressed in low expressing cells compared to high expressing cells in breast cancer, was most of them were related to different pathway, and the one that was coming on top was with Wnt pathway. And it was known from literature that USP19 was regulating the expression or the levels of LRP6, which is a co-receptor of Wnt pathway. And this was already published when you take out basically LRP6 is ubiquitinated. If you have USP19, USP19 is getting rid of the ubiquitination, and therefore the protein is increased. If you remove USP19, the ubiquitination happens much faster, and the protein goes down. So basically, what we observe is that when we knock down USP19, the levels of LRP6 were decreases. And if we were looking in the cells where we did the complementation, when we reintroduced USP19, the levels of LRP6 were going up compared to the levels and similar to the levels that we got in the overexpressing in the very fast migrating cells. So we also observe a rescue phenotype in terms of when as I told you, we have these cells that couldn't migrate. When we introduce the wild type, when we introduce the wild type version, we're able to migrate. And what we did is knock down LRP6 in those cells. And basically what we observed is that when we knock down LRP6, the cells that were able to migrate, now they lost their ability for migration. So confirming the fact that at least partially the phenotype that we're observing in through the effect of USP19 on migration was due to the modulation of the co-receptor LRP6. So then what we did is we did a collaboration with a group. First of all, we did two things. We did a collaboration with a group in Chieti, Vincenzo De Laurenti, and we did a study staining patients' samples to see the expression of USP19 in different tumors. Because the fact is that when we try to do a bioinformatics on USP19, although we observe as a general rule when you pair expressing or low express, high expressing, you can find some difference. We're not basically able to find many difference in terms of effect on the survival. And most of the time, the fact is that genes related to ubiquitination are not highly controlled at the level of transcription, but rather they are controlled at the level of protein. So we couldn't find in the databases a lot of data regarding protein levels. Now there are much more, and we were repeating the analysis, but at the time, as there were not that many, we decided to do our own studies, staining samples from breast cancer patients, samples taken from breast cancer patients, and looking at the levels of expression of USP19 and the relation to the overall survivals and the free survival period. And basically what we observed is that regarding disease-free survival or overall survival, there was no difference in cells in samples obtained from patients overexpressing or with overexpression of USP19 compared to underexpression or no overexpression of USP19. But we observe a difference when we analyze as a endpoint distant relapse-free survival, meaning the chances that the patients that have USP19 overexpression would result in metastasis in the future. And actually, what it seems that USP19 is a prognostic predictor of metastasis in breast cancer patients. That's what we found out. Then we decided to take a little bit further, and we did another screening where instead of just taking the cells and making the screening in vitro, we took the cells that that came out from our screening and directly injected inside the mice. And the mice, by tail injection. This time, the experiment I showed you before, it was a cell line where we knocked down USP19. This is taking out the bulk of the population and injecting there, and basically finding which gene was present was absent in the metastasis compared to the control cells. And other than finding USP19, we find other genes, and the other genes that we found and we focus our attention was HER1. HER1 is in this case, it's not a deubiquitinase, but it's an ubiquitinase. It's something that ubiquitinates. And similar to USP19, when we knock it down, the mice, we couldn't produce the injection in the fat part of the mice didn't produce a tumor formation, and metastasis was highly reduced when the overexpression of USP19, of HER1 was obtained by shRNA. So to summarize, what we show is that by targeting motility, we were able to find genes that somehow were important in the survival of the tumor cell, not in a 2D condition, but in a 3D condition, which resembles more what happens inside the tissue. And interesting, there is a biotech called Alma that they were working on developing inhibitor of DAPs for a while, and they developed an inhibitor molecule that is inhibiting USP19. And when we contacted them, they told us that they had abandoned the idea of following up in cancer, because what they saw is that when they were treating cells with these inhibitors, the cells were alive. And we told them, okay, we know that, because if you are treating the cells in the Petri dish, the ESP19 is not required for survival, but we have quite strong evidence showing that if you treat the cells in a more where 3D constraints for their survival are in place, you have a huge impact with the inhibition of ESP19. So we pair with them, and they send us the inhibitor. Is they develop a second version which is biorally available, and therefore we are doing an experiment to prove that not only as the genetic approach work, but possibly also the pharmacological approach might work. Because many times, again, especially with DAPs, with the ubiquitinases enzymes, people have not been very successful in developing inhibitors. So this is was a strike of luck to find this biotech that now they are pursuing the function of ESP19 in a completely different setup, and we are trying to push forward to see whether we can reproduce the data with a pharmacological inhibitor, and for that we are doing a collaboration with a group in Sao Paulo that will help us figure it out. So, and last for this part is that working with other E3 ligases. In the past, I was working with an E3 ligase that was related to the cell cycle control, and that we proved that it was important for cell cycle exit. And Juliana, who is now also in collaboration with Marina Vaccaro from the el Instituto de Biología Molecular at the Medicine faculty in Buenos Aires. She did an experiment proving that there is a connection between, or further substantiate this connection I told you about ubiquitination and autophagy. And lastly, she now moved to Verona, and she's working with Daniele Vaccaro, and with them, what we did is study another E3 ligase that was mediating the degradation of another substrate involved in tumor growth and cell migration. So now, moving towards the new things we are doing, unfortunately, there is a lot of things we are doing, but we don't have. It's a different field. It's a new field where I'm heading. So I'm basically telling you more about what is the strategy or how I see data science in this aspect, and I'll tell you about the projects that are ongoing. So we are discussing with Tito the fact that people now, everybody now is talking about artificial intelligence, but basically when most of us are referring to artificial intelligence, we're meaning about machine learning, meaning these algorithms that they can learn from experience and they can predict something with accuracy. So, but we don't have to forget that behind all of that, there is all good statistics, and it's just other ways of inquire the same data. So the difference between statistics and machine learning basically is that with statistics, you can explain, you can infer things that happens inside the, it's more complex, but if I have to define it in a nutshell, with the statistic, you can infer about the behavior of the population you're studying, while with machine learning, you can predict. And sometimes you can predict, but you cannot really understand why the prediction is working okay. So nowadays, there are many other systems that that are helping you to understand the black box system of machine learning. But the important thing is that the problems that are difficult are always difficult also to solve with machine learning. So it's not just a magic wand that you use to solve everything. But where I see that is most interested is when you start in a complex system. No, a complex system is not, I mean, a simple system is something that with a equation you can predict the movement and where it's going to be in the time, in any time that and you can predict all the behavior. A complicated one is when you have a lot of things to do. Flying a 747 Boeing is a very complicated thing, but it's still, if you know all the knobs and twitch that you have to do, you can do it. Chaos is on the other side, and there is there are part of science that is dealing with that. But complex is a system where you realize that there is a pattern there, but the pattern is not easy predicted or identified. Knowing where a flock of birds will turn in the next second is not really predictable, and patients or any living organism is a very complex system where I guess that machine learning can help a lot, at least to produce some hypotheses that then we have to validate in the old way. And the important things that we have is, and this is just a word of warning, is that when the data you put inside is not good, whether you're taking out is not good. So this is really, and there is no really an interesting or a very robust way of checking the quality of the data. Because even if with statistics, you can check if all your data is normal, if it is obeying to all the things that they need to obey. But here is a little bit more complex, and sometimes it's not easy to know whether your data is good or not. And you may end up by developing things that are learning a lot about the data you have, but it's only useful for the data you had. And then when you try to apply for new data, sometimes you have an effect of overfitting, meaning that the algorithm learn a lot about the data you used to train, but then it's not able to catch the generalities of the system. And the other problem is that most of it, just the fun part is almost at 10% of the time. Most of the time you're just spending moving data from one place to another. And for doing this, this is by definition, is a multidisciplinary endeavor where you need not only to have people that know how to press the button, then you need to know people how to deal with numbers, and most important, you need to have the person that knows about the problem. So most of the time, what I've been doing in the last years is dealing with in the interface between the physicians and the engineers, that when they talk to each other, it's just, although they can speak the same language, but they don't understand. So what I've been doing is I've been involved in imaging analysis, self-segmentation of slides from myocytes. We have dealing with data from patients, from different lab results, trying to make a better predictor of renal function deterioration. We also try to have we dealt with a very huge data set where they were dealing with all the lab reports from a couple of years and trying to figure it out whether there was a way to estimate some parameters and prevent to overstudy patients. Also, more recently, I've been dealing with a project where what we decided to do is that here, during the first trimester of pregnancy, they do a test blood, and then they do that on the first trimester, second, and the third one. And pregnancy, the gestational diabetes is the one that you get through the when you are pregnant, and then you can. It's not that you are diabetic, and then after the pregnancy you can go back and being and having no symptom of diabetes. Is an interesting disease, and what we try to do is using the tests that they are taking from the first semester, the first trimester, where the diabetes is diagnosed at the second trimester, and build up a machine learning algorithm to predict whether the patient will develop or not diabetes. And we obtained quite an interesting data, but this project in particular, and the approach I'm trying to use, is that the idea is not having as an endpoint, but as a starting point. Most of the time, through my career, we started from the genes, from the interaction. We were doing a lot of experiment and trying to figure it out, what was the biological or physiological relevance. This would be to do the reverse approach. In other words, is making the paper from figure 8 to figure 1 instead of from figure 1 to figure 8. So using machine learning to inquire, to produce and analyze big set of data, and then if anything comes out, then we have to figure it out how to translate that into solid, understandable information that can be used both for clinician, but also you can take it back and make it a molecular understanding of the process more accurate. So with that, I will finish, and I will be very happy to take any questions. Thank you very much.